Did I?
Oh dear, I've got a slip disc. Oh, I've got a slip disc.
Hello there, it's the fourth video in the Evolutionary Ecology series, and today we're going to be talking about possibly one of the more mysterious but exciting areas of evolutionary ecology, and that is speciation, the production of new species. And we're going to be taking a look at it from a genomic point of view. Now, I've talked about in a video ages ago about different species concept, but for the context of this video, we're going to be talking about the biological species concept. If you want to know what that is, I've put a link to the video in the description, so you can go and check it out, which is all very nice. So I've written out the various stages of this genomic approach to speciation that has been proposed before, so let's see if we can make sense of it, shall we? So, it's been suggested that speciation can start by divergence at a single locus in the genome. When two populations get separated, a single locus diverges. Now, as those populations become separated for long enough spaces of time, then they begin to bring other genes with them through this process of genetic hitchhiking. So they could be physically linked with it or in linkage disequilibria with it. So they'll be carried along as a result as well. So what you've got is basically a chain reaction happening. Because with that, then you can get divergence of whole genomic regions, whole areas of the genome will diverge from each other in these two populations. So then eventually you'll get the divergence of whole genomes. So speciation has taken place, you've got two new species. As I'm sure you know, there's various different types of speciation. So there's allopatric, peripatric, parapatric, sympatric. Okay, we're not going to focus so much on these individual definitions in this video, and that's because I've talked about them before somewhere else. Okay? So it's kind of assumed that we already know what we're talking about here. But once again, I've posted the videos to those videos in the description below as well. And this model which I've just described is relevant to any one of these forms of speciation. Now, as we'll see later, sympatric speciation is without doubt the most controversial. So this is where variants are produced within one range, and these different variants become species eventually by adapting to different parts of that range. Okay, but we're going to be looking later at the end of the video that there is some evidence for sympatric speciation. Now, for two populations to become good species, if you like, there's got to be reproductive isolation. That's one of the main parts of the biological species concept. Okay, so as I've talked about before, there's pre-zygotic and post-zygotic reproductive isolation. Pre-zygotic reproductive isolation could be anything to do with sexual selection, so sexual signalling traits, that sort of thing, barriers to dispersal and gene flow, differences in life histories, all that sort of thing. Okay? Whilst post-zygotic reproductive isolation is all about hybrids being infertile. Actually, it's also worth noting that hybridization can actually produce species as well, particularly in plants. If two plants hybridize together, there's a possibility that one of the offspring produced will be able to survive and germinate, and there you go, hey presto, a new species. Okay, so it can happen. Olnick and Neer in 2005 looked at this hybrid incompatibility in American centrichids. These are the bassers and the sunfishers, okay? And a phylogeny was produced using fossil evidence to see phylogenetic relationships between particular species. And they then tested this hybrid compatibility. And it was shown the longer ago that the species diverged, the less compatible the hybrids were. However, what is amazing is that if the divergence was as long ago as 10 million years, the success rate of hybrids was pretty high, and some of those hybrids actually turned out to be fertile. So this shows that the rate of post-zygotic genomic incompatibility is pretty slow really if it takes 10 million years. Now that last model we talked about, describing a genomic approach to speciation, was very good at describing how it might happen, but it didn't explain why it might happen. And that's exactly what this next model demonstrates. So let's try and follow this step by step then. Well, it starts off with a polymorphism evolving, which conveys differences in ecological preference. Okay, so there's some divergence in some gene, let's say, which conveys a preference for a particular ecological location. Okay. Now, because of that, that'll lead to assortative mating, because these organisms are only likely to mate with organisms in their local ecology. So then what you get is this ecological preference and mate preference becoming linked in some way. Now, they could be physically linked in the genome, or the same gene could be having multiple effects. And this is what we call pleiotropy. Pleiotropy is where one gene affects multiple traits. So it could be this locus that we talked about in the original model displays an ecological preference, but also a mate preference. This is without doubt the hardest part to explain, because it's very difficult to test for it. But once this has occurred though, then selection against the opposing population can be complete. So then you get speciation. Okay, so that's a theory. Are there any examples that we can talk about then? Well, of course there are. We're going to return back to our Tamamus stick insects, which, as we talked about 2 episodes ago, have 2 ecomorphs which occur in the same habitat. But these 2 ecomorphs are adapted to different host plants, and they differ in body width and stripe length. What Nozzle et al. Did in 2002, the same guy who did the studies that we talked about last time, he got loads of these stick insects from different populations and put them with each other, and he measured to see whether they mated or not. And what he found is that individuals are more likely to mate with an individual from the same population. But it wasn't just as simple as that. What he actually found is that there was a hierarchical mate preference. Okay, so the individuals which the stick insects preferred to mate with next were individuals from different populations but from the same host plant. Okay, so that means they had very similar morphologies. So therefore, right at the other end of the spectrum, the stick insects which they like to mate with least were ones from a different population and that live on a different host plant. Therefore, reproductive isolation isn't complete because you can still get mating between these populations, but mate preferences do certainly seem to be evolving over time. Now, one of the more famous examples when studying speciation comes from this little boring fly, okay? It's the apple maggot fly, Ragoletis pomonella, which is something which I believe I've also talked about before in one of the videos, so check out the videos in the description below, basically, to boost my views up. To learn more about Ragoletis pomonella. So what these flies do is that they lay their eggs inside a fruit, and inside that fruit the maggots will develop, pupate, and then they'll emerge as flies the following year, so the cycle can start again. Now, until the 1850s, all Ragoletis pomonella flies laid their eggs on hawthorn. But once apples were introduced, the flies started to lay their eggs on it. So that got the scientists questioning whether actually we've got 2 species evolving before our very eyes. So first thing you can do then is do some behavioural experiments, and that's exactly what Lin et al. Did in 2005. And what they did was they exposed these flies to a mixture of blends, either a hawthorn blend or an apple blend, very tasty. And they looked to see whether they could locate them purely due to olfactory cues. Okay. And what they found is that flies that laid their eggs on apples located the apple blend much more efficiently, and vice versa for the hawthorn blend. But it wasn't like the hawthorn flies couldn't detect the apple blend. Some of them did reach it. This means they could potentially mate with the opposite morph and produce viable offspring. Okay, so maybe they're not two different species, yet anyway. But there's also a mating incompatibility in terms of the timing in which these fruits emerge. There's about a month's gap in between the timings of hawthorn and apples being produced. And even if hybrids were produced, that hybrid probably would be flying at the wrong time for it to lay its eggs on either hawthorn or apple. So it would be pretty stuffed, basically. So what's interesting here is that the gene, whatever it is, for fruit preference, also determines whether populations can mate together. So therefore, we've got a gene which has multiple effects. It's pleiotropic. And we call these pleiotropic genes magic traits, which is quite a nice name, I suppose. And this was backed up by a study called Philcack, Philco, nice name, Italian 2000, where he concluded that hybrids can't locate plants very well. And as a result, of course, they're selected against. So maybe Ragoletis pomonella is on the way to becoming two different species. Right, so magic traits, that's a pretty nice word, isn't it? What actually do we mean though? Well, magic traits are any traits which are subject to divergent selection whilst also contributing to assortative mating. Now, this could be due to pleiotropic expressions of the same gene, as we've talked about, or it could be two closely linked genes. Okay, so for example, genes that could be next to each other on the chromosome or something, things which are physically linked. And as we've talked about, these ecological preference and mate choice genes are likely to be linked pretty early on in the speciation process. Okay, so an example relating to that are these Lord Howe Island palms. Right, there are two species. There's the Kentia, I think that's how you say it anyway, and the curly palm. And these two are found in Sympatric. And they're thought to have diverged around 2 million years ago. Now, the mechanisms of this speciation are thought to be due to differences in flowering time, in reproduction time. But this isn't due to altitude or anything. This is thought to be due to soil pH. And this difference in soil pH will then, as a result of that, affect flowering time. Now, you've got to remember, it's quite difficult measuring something like a Lord Howe Island palm, because obviously they take years to grow, so it's difficult just to get some Lord Howe Island palm seeds, put them in the lab and see what happens, because it would take ages. So we can only infer things from these kind of studies, or using these kind of organisms. Okay? But it's likely that this genetic polymorphism for substrate type, or soil pH, is what triggered this divergence, because as a result of that, it affected flowering time. So whatever that gene was which affected the flowering time and the preference for the substrate type, then that is definitely a magic trait, but we haven't exactly located it yet. As well, with many of the studies we haven't done yet. Now, we've talked about one model for why this divergence at the genomic level may take place. Another model was proposed by Van Doorn et al. In 2009. Now, if we think back to our original model, we talked about how a polymorphism arose that affected ecological preference. Okay, this is slightly different. This is suggesting that a polymorphism arose which conveys some sort of ecological difference. Okay, it made different individuals better adapted to different environments, and as a result, adapted individuals have higher fitness. Now, it's this adoption of these dichotomous habitats which means immigrants coming from another habitat are less likely to invade because they're not as well adapted. So therefore, adapted individuals are likely to be healthier. This is going to help in sexual selection and mate choice, so that means they're going to reproduce more, and as a result of that, selection against the opposing population will be complete. And of course, when generalists are reasonably fit, then we shouldn't get any speciation taking place at all. The model fails, basically. You can theorise on how that model might apply, but there's been very little literature experimentally, specifically testing for it, which is a bit of a shame. But well, that's what the future holds, isn't it? But Sympatric Speciation, on the other hand, that's also a pretty controversial topic. Can it happen? Can it not? And this is where we're just in time, pretty much. A paper by Belinsky et al. In 2016 has produced one of the best pieces of evidence of sympatric speciation occurring that there's ever been. Okay? And this is a study on cichlids. Again, I told you there were loads of fish. On Lake Masoko in Tanzania. Within this genus of Latvia Cichlids, we have two morphs. Okay? The first morph tends to live in shallower water, and the males are yellow with red heads. Okay? Whilst the other morph tends to live a more benthic lifestyle in the deeper water, and these are blue. Now this is a very deep lake, by the way. It's a volcanic crater lake. And other differences we see is that the shallower morphs tend to have different diets. So they tend to feed on algae and hard-shelled prey, whilst the deep water morphs feed on zooplankton. And we also see differences in morphology between these morphs. There's differences in body shape, mass, diet, and pharyngeal jaws as well. So there's loads of instantly noticeable differences, even though they're the same species. Okay? And we're pretty sure that they are the same species, because as far as we know from constructing phylogenies, that there's been no double colonisation event of Lake Masoko from these cichlids. So they're a monophyletic group, basically, and this was done by sequencing the entire genome of around 100 individuals, and they did find that all the ones from Lake Masoko appeared to be grouped together in a monophyletic group. And it was also shown that these benthic forms are likely to be the individuals that budded off. This is thought to have happened around 1,000 to 5,000 years ago, so pretty recently, really, and they budded off from the shallower morphs to go for a more benthic, deep water lifestyle. And this was done by looking at genomic similarity, and the actual time of colonisation of this lake was calculated to be around 10,000 years ago, which is quite handy because it fits in nicely with when we think the lake was formed, which was around 50,000 years ago, right? So it all seems to be fitting in pretty well. Now, the next step to do then was to see whether there was divergence at particular genetic loci. So what we did, well, not me, I was never there, what am I talking about? You work out FST values. Now, FST is a measure of divergence at a particular point in the genome. And what you see is, if you scan, remember this was done using whole genome sequencers, okay, so very high tech. If you look across the genome, you don't see many divergences, but you do see peaks in certain genes which appear to be diverging. Now, we're going to have a whole video next time on looking at how we can identify these adaptive genes that lead to adaptive radiations and so on. But in this case, these genes that are identified seem to be involved in things like vision and skeletal morphology. And they also show to be very tightly clustered in the genome. There are areas of the genome where you see really high, loads and loads of peaks, one after the other. And within these peaks, we also see peaks in genes which aren't necessarily that related to things like vision and skeletal morphology, but they've also just been carried along because they're physically linked to genes that are. Okay, so you get linkage here, linkage disequilibria, and as a result of that, there's been genetic hitchhiking. Okay. So we've talked about how these highly divergent regions, these HDRs, are involved in these sets of genes. So it's these highly divergent regions of the genome that we can call speciation islands, and 98 of these were identified in these fish after looking at the whole genome. And these genes as I say, are associated in all sorts of metabolic processes. They're involved in morphogenesis, hormone signaling, sensory systems, cytoskeletons, and protein translation. So what you've got here is a really nice overall study, not only looking at differences in morphology, but going right down to individual genes and seeing how much they're diverging. So it's quite remarkable, really. But what we've got to learn from this is that maybe this is so good because these cichlids are pretty early on in the stages of speciation. Okay, we've caught them at just the right time because, as we've said, it's only been 2,000 to 5,000 years since they started diverging. That's no time at all. Okay. Similar studies have been done in corvids and they've shown similar things. In terms of the Tamema stick insects, we see less of these speciation islands, but maybe that's because these Tamema stick insects are further on in the speciation process. So if we go back to that original genomic mechanism of speciation where you get divergence at particular loci, then you get genetic hitchhiking, then divergence of whole genomic regions and so on. They're already at the later stages of that. So you get divergence across the whole genome. Here, divergence hasn't been taking place for a very long time, so you only get divergence in the particular genes of interest. So the ones involved in vision, skeletal morphology, that sort of thing. Okay, so when we're studying speciation, the thing we've got to learn here is that it's good to study species which are only just diverging. If you look at two completely distantly related species, it's hard to learn much from that because so much time has gone past that the genomes have already had loads of time to evolve and change. Okay, right, that's all I've got time for today. I'll be back next time, as I said, looking at functional genes. Okay, see you then.